Assalamu alaikum. I am Rukhsana Begum, Associate Professor in Botany, Government Girls Degree College, Zamzama, Karachi. Dear students, today we will discuss the life cycle of Angiosperm. Belongs to subdivision Spermopsida, the flowering and seed plants. Chapter number 9, Kingdom Plantae of first year Botany. The seed plants are divided into two groups, Gymnosperm and Angiosperm. Gymnos mean naked, sperma mean seeds. They have naked seeds. Ovules are not covered by ovary. Angion, in Angiosperm, angion mean vessel or cup. Sperma mean seed. Their ovules are enclosed in ovary. As our topic is concerned with flowers, so let's have a look on flower. A flower is highly modified form of stem bearing sex organ. So they are associated with reproduction of plant by producing seeds. It is produced at apex or tip of stalk called pedicel. Just above the pedicel, there is a swollen part which is floral axis called thalamus or receptacle. And the second whorl of flower is floral leaves. The floral leaves are 4 in numbers and they are inserted in the thalamus. The first whorl, which is green in color, that is called calyx. The second colored part or whorl is called corolla. The third, the male reproductive organ called androecium or individual member is called stamen. And the fourth one is female reproductive organ is called carpel or pistil. Now let's come to the topic, the life cycle of Angiosperm. The life cycle of Angiosperm is divided into two stages. The first stage is called sporophyte, which is diploid, and the second stage is called gametophyte, which is haploid. The sporophyte stage, in this stage, two types of spores are formed. Microspore or pollen grain produced by microsporophyll or stamens. The megaspore or eggs are produced by megasporophyll or carpel. Now the microsporophyll or stamens, the male reproductive organ. It consists of anther, filament and connective. The anther has 2 lobes. Each lobe contains 2 chambers called microsporangia or pollen sac. Each sac contains million of diploid microsporocytes which divided by meiosis and produce 4 haploid microspores. In this way, numerous microspores are formed in each sac. The wall of each microspore thickens and the nucleus divides mitotically, producing a generative nucleus and tube nucleus. The thick-walled 2-nucleate cell is pollen grain, a male gametophyte which is released from the anther when the mature anther split open. Now the megasporophyll or carpel. The carpel consists of 3 parts, the stigma, style and ovary. An ovary has 1 or more chambers called locules. In each locule, 1 or more sac-like structures are present called ovule. The ovule contains a large diploid megasporocyte which divides by meiosis and form 4 haploid megaspores. Out of these 4, only 1 survives and 3 are disintegrated. The microspore and megaspores are heterosporous. Now come to the development of female gametophyte. Before fertilization, many changes occurs in the ovule to form female gametophyte or egg. The functional megaspore, which is haploid, its nucleus divides into 2 nuclei. These daughter nuclei separate from each other and move to opposite poles. The nucleus at the upper end divides twice by mitosis to form 4 nuclei. Similarly, the nucleus at the lower end also produces 4 nuclei. Now, 1 nucleus from each of the 2 groups is migrated to the center of the embryo sac, where the 2 nuclei fuse together and form polar nuclei or definitive nuclei. As the matured embryo sac contains 2 groups of 3 haploid cells at each end, a group of cells towards micropylar region is called egg apparatus, of which 1 cell is comparatively larger called egg, whereas the remaining 2 are synergids. Another group of 3 cells lying at opposite pole towards chalaza region are called antipodal cells, which have no function. A large distinct diploid nucleus is present in the center of the embryo sac is called polar nuclei or definitive nucleus. Now let's discuss the ovule. Or as we see here, there are different layers and other parts are present in the ovule. So we are here discuss an ovule. The structure of ovule, we can say. Each ovule is an oval structure which is attached to the placenta, a mass of tissue on the inner wall of ovary, through a slender stalk called funicle. The point of attachment of the ovule to its funicle is called hilum. Inside the ovule, there is a mass of tissues called nucellus, surrounded by inner and outer integument or layers. A small opening or pore is present at the apex of an ovule, just above, opposite to funicle, called micropyle. It is pore through which pollen tube enters into the ovule. The tissues lying in between funicle and nucellus, where all integument merge, is called chalaza. The nucellus near to micropylar end has a large oval structure called embryo sac, which contains 8 cell structure, or we can say the egg. Now let's discuss the development of male gametophyte and germination of pollen grain. Each microspore or pollen grain or microgametophyte, which is haploid, it consists of outer coat, exine, which is thick, and inner thin wall called entine, and a nucleus. Now this microspore, when reaches at the stigma, the nucleus of the pollen grain divides into 2 by mitosis. The generative nucleus, which give rise to 2 male gametes, and the tube nucleus, which give rise to pollen tube. Due to stimulation occurs at stigma, the internal wall of pollen grain protrudes out from germ pore in the form of small tube called germ tube, which carries 2 male gametes. It passes through the style of carpel and reaches the ovule inside the ovary. Now, when these gametes reaches inside the embryo sac, the 2 male gametes reached at the micropyle in the ovule, as I say, one fuses with the diploid definitive nuclei or polar nuclei and form triploid endosperm nucleus. The other male gamete fuses with the haploid egg to form a diploid zygote. The whole process is called double fertilization, which is a characteristic feature of angiosperm. The zygote gives rise to embryo. The embryo consists of embryonic root and cotyledons. The endosperm nucleus will form the endosperm, food storing tissues, which supply food to the developing embryo. Now the ovule, which contains embryo, is converted into seed. The fruit is developed from ovary. The seeds are protected in the fruit. When the seed germinates, it gives rise to seedling, and from seedling an adult or mature sporophyte is produced again. In this way, the life cycle is completed. Thank you.